I'm recording. I'll record today so that you can go back, because it might be something that you like watch and pay attention more than anything, and then go back and fill in the blanks, because it's harder to take just notes off of a process. So you may want to watch and follow along. So if you get through today and you're like, okay, yeah, I was able to follow along, then go back and fill in the gaps. And so I am going to record this. I will put it a link to it later. It might be tonight, but I'll put a link later to it in Schoology folder. Okay. So I'm going to go ahead and share my screen in a second, but first, let's talk about DNA replication. DNA is replicating. You start off with one DNA molecule, double-stranded, double helix. Think one spiral staircase. The end, we should end up with two, correct? Isn't that what replication means? To make a copy, okay? Don't overthink, don't overanalyze that part. So DNA replication, think back to cell division, think interphase. In interphase, there are three major stages. What are the three major stages of interphase in cell division or before cell division?
G1S, G2.
G1S, G2. G1, the cell grows, organelles copy. What happens in S phase? S stands for synthesis. What happens in S phase? Someone who has an answer? Erica said the DNA replicates, the DNA copies. So when we said in the S phase, the DNA copies, that's what we're going through. And I told you it takes about 6 hours to do. One cell, skin cell, becomes two in about 20 hours. 6 of those hours is just copying the DNA. So what we're about to do today takes, in actuality, about 6 hours, okay? So let me show you the process of DNA replication. And if at any point in time you want to take a picture of what I'm showing you, you can, and I'll pause to let you do that. So then you can look at the pictures later as well, okay? So let's say that this is a portion of my DNA molecule. In actuality, you know, this is one base pair, second base pair, third base pair. Between these two, what type of bond exists?
Hydrogen.
Hydrogen. And between C and G, how many hydrogen bonds are there?
3.
3. So I could go ahead and just show you that and say, okay, there's my 3 hydrogen bonds. Adenine and thymine have 2, and you would see that. You also will see that I've left the phosphates and the sugars off, but I did label the ends 3' and 5'. So here's 5, so then the bottom has to be 3. This is 5, that has to be 3. Doesn't this show anti-parallel by me just labeling the ends, right? Okay. Now in the 5', just to show you one, there really would be a sugar coming off of this and a phosphate up here. There's one nucleotide. Here's another nucleotide. And I could continue that on. So they call this the phosphate sugar backbone because it's just phosphates and sugars. Over here, you would see phosphate and sugar, but you would see it upside down with respect to its match. Remember, in 5', carbon 5 of the sugar is sticking up. Basically, the phosphate is sticking up, okay? So carbon 5 would be about there. And so I could continue this process, but the paper would get pretty cluttered, right? And so all I'm leaving off is the phosphate sugar backbone because it's phosphate deoxyribose, a base, phosphate deoxyribose, a base, phosphate deoxyribose, and a base. So I've simplified by just leaving the phosphate sugar backbone off. Is that fair enough? Because the only thing that's differing are the bases. So I'm just showing the bases, but understand it's a whole DNA molecule. So this is showing a portion because in actuality, your DNA in one cell is about 6 feet long. 3 billion base pairs in one cell. I showed you about 14, 15 base pairs. So imagine this really long, and we're just replicating this one part right now. Now, let me ask you something. If I laid down a hypothetical DNA molecule in our classroom, from the front of the classroom to the back of the classroom, and I said, make a copy of this, I don't care how you do it. Rather than one of you or two of you working at one end and saying, okay, I got to make a copy of this, I got to make a copy of this, I got to make a copy of this, and continuing on, wouldn't it be better if a bunch of you got together and said, hey, I'll start up here, you start here, and I'll start down there, and then we'll meet up? Wouldn't that go faster? Wouldn't more people doing the job make it faster? So imagine your 6 feet of DNA. This is one portion. Somewhere else on the 6 feet, other people are working. Somewhere else on the 6 feet, other people are working. And eventually you'll all meet up. So this is the portion that you are working on. So imagine a double helix going in this direction. Imagine a double helix going in the bottom direction. Okay. Do you understand the setup of my paper right now, like what I have written on here? It's a portion of a DNA molecule, and we're going to show replication of this portion that you're responsible for. Okay? All right, so this is my DNA molecule. In the end, I have to end up with 2 DNA molecules. Our goal basically is to get to this. We start with 1, we end up with 2. Isn't that DNA replication? Okay, so we want to go through the steps to get you there. So we're going to start off with what would be our original DNA molecule. It's like when you go to make a photocopy, don't you take an original with you and put it on the copier and make a copy, right? So this is what you're taking with you. These 2 strands that you are starting with are called the template strands. Okay. We're using that as a template, just like the paper you would take to make a photocopy of is your template, okay? So it's sort of the original. All right, so we have our template strands here. We're gonna use this as a template to make a new DNA molecule. I just labeled these 5' and 3'. Originally, you wouldn't have known, but now that I put the phosphates and the sugars on here, you can see why it's 5 and 3, okay? All right, so there are a bunch of terms that are going to come up, okay? Now, what we need to do is basically pull these strands apart because you're going to use these as a template to make a copy. If I gave you just this one strand of DNA right there, couldn't you make the complementary strand off of it, right? And if I gave you this strand right here, couldn't you make the complementary strand off of it? So we're going to use these 2 as our template to make new strands. So what's going to happen is there's going to be an enzyme that's called topoisomerase. Okay, so there's a lot of terms coming up. How do you know it's an enzyme? Topoisomerase. How do you know it's an enzyme?
ACE.
The ASE ending, okay? Now, the enzymes are all the workers, okay? So I want you to take a look at this DNA molecule, this red and black pipe cleaners, okay? You see that there's 2 strands wrapped around one another, right? They're held together. Now imagine this is your 6 feet of DNA, and you're working, you started, somebody's working at this end, somebody's working here, somebody's working down here, and you happen to be working, like, let's say right here in the middle. So what you need to do is pull these strands apart so that you can work with them. Temporarily pull them apart. But look how tightly it's wound. So when you try to pull apart, like I'm pulling really hard right now, and it's really hard for me to separate these 2 strands, okay? So what topoisomerase does, it alleviates some of the tension by unwinding a little bit. So I can unwind a little bit, and it might look like this, where now you can get in there and you can actually start working because you can separate the strands a little bit. And so you can work on this red one, the red one and the black one, okay? So topoisomerase alleviates some of the winding, but you notice that it's still a double helix where it's not being worked on. So you don't completely separate the 2 strands. You don't completely separate the 6 feet. You just separate the part you're working on at that time. So look how wound up this one is compared to this one. Okay, so topoisomerase works to help alleviate some of the tension so that you can separate the strands while you're working. Okay, so I'll put topoisomerase up here, okay, just so that it can start to alleviate. Okay, so there's my topoisomerase. Now what we need to do is we need to separate these strands so that we can work on them. And so there's an enzyme called helicase. Helicase, okay? And helicase will unwind and unzip. So it is the one that actually comes in here and unwinds and unzips, separates. Think of the two strands of DNA like a zipper, and you pull your zipper down. Doesn't it separate the two strands then? Okay? And you stop me when you need to, okay?
So helicase is the one actually separating them, but then the other one is just loosening it.
Yes, topoisomerase just loosens how wound up it is. Okay, so topoisomerase would do what I'm doing right now by like just loosening. Doesn't completely unwind, just loosens. Okay, and then
what's the template strand again?
Separates. Say it again.
What's the template strand?
Template strands are the original strands you're going to use. How do you make
all the 6 feet?
Say it again.
Like all the 6 feet, or just that little portion?
The whole 6 feet will be.
Oh.
Okay. The whole 6 feet will be your template strands. The template is what you're using as a guide to make something new. If I don't give you these strands and I don't show you this picture and I say, make a copy of DNA, and you're like, well, how do I know where A's and T's and C's and G's go? You use this and now you can make a copy. So that's why these are your template strands. It's the original strands you start with. Okay. So helicase, let's just say we're starting down here. Okay. It really doesn't matter because eventually you're going to get it all copied, but we'll just start down here. So what helicase will do is it will come in and it will move through and separate the strands of DNA. This is my helicase, okay? So it pushes through. What's it breaking through?
Hydrogen bonds.
Hydrogen bonds. So it broke through. It's this big guy that breaks through so that these two strands, even if they try to go back together, they can't go back together because helicase is blocking it. Okay, so it's this big giant enzyme. Okay. Topoisomerase is still up here, just alleviating some of that stress and tension. Okay. So now we have space to make our new DNA. These, by the way, are all the terms that are going to come up today. So again, this is being recorded, so you'll see. So when you go back, or if you want to take a picture at some point, you can. It's all up to you. Okay. All right. So single strand binding proteins. Single strand binding proteins. Okay, single strand binding proteins. Guess what these proteins bind to?
Single strand.
Single strands. Isn't this a single strand of DNA right now? The part that's separated? Isn't this another one? So what single strand binding proteins do, and if you can see, I just put like SSBP for single strand binding proteins, okay? Single strand binding proteins are going to hold the two strands apart. So it's as if they're basically like grabbing the strands and holding them apart so that work can be done. Okay? So they're going to hold the strands apart because naturally the strands want to try to go back together. So they're going to hold them apart so you can work. Are we good so far? Anything so far? Okay. So helicase push through. It'll go little bits at a time. isn't it always better to do things like slowly so that you're more effective rather than rushing through? So in order for this replication to take place, this is DNA. You make a mistake and it could be a mutation. It can lead to a genetic disorder. It could be a big deal. Go slowly. That's why it takes 6 hours. So helicase pushes through a little bit. I just chose the location it was going to push through. There's no exact. I showed 5 bases it pushed through here. In actuality, it could be 50, 100, 1,000. You know, your number, you remember, you have 3 billion base pairs. So there's nothing exact. It just pushes through a little bit at a time, okay? So where the helicase stops is called the replication fork. Isn't it like a fork in the road? Okay, and aren't we doing DNA replication? So it's called the replication fork, okay? Now you have space and some work can be done. There's an enzyme called DNA polymerase III. There's the fact that there's a DNA polymerase III probably should tell you that there's other DNA polymerases. Okay.
Dr. Brooks,
Yes.
sorry, did you say the replication fork was the space in between the?
The replication fork is a location where helicase stops.
Okay, thank you.
Okay. So DNA polymerase III. This is going to be my DNA polymerase III. I'm using what I have. Okay. Is that fair enough? DNA polymerase III. Okay. DNA polymerase, all of them, their job is to make new DNA strands. Their job is to add DNA nucleotides. They're really the ones that are making the new strands. All right, so we're using these strands as a template. So DNA polymerase III. Over here, let's go on this side. New strands are always made from the new strands 5' to 3'. Okay, say that to yourself. New strands are made from their own 5 to 3, 5 to 3. If this end of our template is 3', and we're going to make a new strand on the inside here, what would we label this end of the new strand that hasn't been made yet? What will it be? Megan, I heard you word 5. Okay, so this would be 5. This strand that hasn't been made yet, what would this end be?
4.
This would be 3. Okay, new strands are made from their 5 to their 3. So let's go over here. DNA polymerase on this side. 5 going towards its 3. So what this DNA polymerase will do is it'll come to here to this first one. It sees a cytosine nucleotide. What nucleotide is it going to add over here when it makes a new strand?
Guanine.
It's going to add guanine, and it's going to go up, and it's going to add cytosine, and it's going to move up, and it's going to add adenine, and then thymine, and then cytosine, and then it's blocked because helicase is in the way, right? Did you see that I'm making the new strand from its 5 towards its 3? And I'm doing it slowly? Did that make sense? Okay. Now over here, we've got a little issue. This is the 3'. So where does this DNA polymerase 3 have to go?
Gotta go up.
It's got to go up because 5 to 3 would be going in this direction. So this DNA polymerase 3 has to travel. The only problem is that it doesn't know where to go. Enzymes may be very smart in the sense that they do all the work, but they're very dumb at the same time, okay? So there is an enzyme called RNA primase. RNA primase, and I'll write it bigger over here. Is going to help DNA polymerase 3 out. This guy is standing here like, wait, this is 3'. I don't know what to do. So then RNA polymerase comes in, And it's like, let me help you out. I will be RNA primase, not polymerase, sorry. I will be RNA primase. And what RNA primase will do is put an RNA primer. So here I am as primase putting an RNA primer in a location to guide DNA polymerase 3. DNA polymerase 3 says, I don't know where to go. RNA primase says, I'll put a flashing light to tell you where to go. Here's that flashing light. So RNA primase puts an RNA primer. And that is what I'm calling that flashing light. Saying, here, come to me. Here's the flashing light. Okay? Let me pause. What questions do you have so far?
So- Sorry.
Why is the primer like just on the left side?
Here, because this was the 5' end, it starts right there. You're going 5 to 3. So it knows exactly where to go. So it's right there, right where it's already standing. But on this side, and you have to imagine this, what if this was 1,000 bases long, not just 5? How would DNA polymerase 3 over here know where to go? It's looking around and it's like, I don't know. Meanwhile, up ahead down the road, it sees this flashing light. So it goes towards that flashing light because it has to travel in the opposite direction, 5 to 3. Does that answer your question? Okay. So now RNA primase, put an RNA primer. This is the primer, okay? This is the flashing light. Now this DNA polymerase 3 is like, oh, okay, I got to go up here. So then it goes up here and the primer's in the way. So then it starts making, where it can, it starts to make the DNA strand going in the opposite direction. It had to travel backwards, didn't it? Okay.
So does the primer block some of them?
Yes, for now.
Okay.
Now.
And it just stays there?
For now.
Okay.
Okay. You'll see where they're removed later. Okay. 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 So at this point in time, now what can be replicated has been, right? What needs to happen next? What should happen next to continue this process?
Don't they have to bond together?
These two will hydrogen bond. So there'll be hydrogen bonds between this C and G. There'll be 2 there. So hydrogen bonds will form here. Hydrogen bonds will form here, okay? But we're doing DNA replication, right? We're still continuing DNA replication. So as these nucleotides, as these new strands are made, hydrogen bonding is naturally taking place. So we want to continue. If we want to continue, then this helicase has to continue moving up. We did the part we could. Now let's go to the next section and do a little bit more. And we just keep doing that. So then if helicase moves up and topoisomerase continues to move up with it, then you get to something that looks like this, okay? Does anybody want to take a picture of this before I move this paper out of the way?
Yeah.
Then can you do it right now, please?
I got it.
Okay. All right. So then helicase moves up a little bit more. And this is where we are so far right now. Okay, this is where we just left off. Okay. Here's our RNA primer. Here are the strands that already have been made so far. Your single strand binding proteins are still working to hold the strands apart. Okay. Helicase stopped. Here's a new replication fork. Now work on the new segment. So here is where we are. What do I do? Let's start with over here. What do I do with this side? What will DNA polymerase 3 do?
Move up.
5 to 3, right? So it's already going 5 to 3. So if we continue, now that it has space, it can keep going. And it will continue moving up, making the complementary strand until it's blocked again. What has to happen over here? Same thing that happened before. So what's going to happen over here?
Primer has to be moved up.
Well, the primer's there. So the concept is right, though. We need an RNA primer, basically, right? Okay. So an RNA primer, RNA primase will come in and put another RNA primer. So let's just say I put it right there. There's an RNA primer. Now this DNA polymerase 3 is like, oh, I see another flashing light. That's where I go next. I've already been to this one. I got to go to this one. Okay. So it's going to start to travel up. So it travels towards the flashing light and the primer's in the way. So it'll make from 5 to 3, it'll make a new strand. Okay, so it'll continue. What happens next? We did what we could so far, so what's next?
Hydrogen bonding.
Well, the hydrogen bonding naturally will take place.
Do we need to remove the primer?
Not yet. Well, we will, but we
Does
still have
helicase
more
go up
DNA
again?
here.
So helicase keeps moving.
So helicase will move up. So if helicase moves up, then we now are at this point where helicase is up here. We've got our single strand binding proteins that are holding apart. We've got DNA polymerase 3s in their position. Here are the primers that were already put in, right? Okay. And so now we can continue on this side. DNA polymerase 3 will just continue up now that helicase is out of the way. And it will just keep making the DNA strands from its 5' to its 3'. What has to happen over here?
Primase.
The primase has to put a primer, correct? Okay. So the primase is going to come put a primer. Let's say that this is the primer. Okay, so I'm going to put it there. And then the DNA polymerase 3 now knows where to travel to jump and then start making the DNA from its 5 towards its 3. So one strand is continuous. One strand, there's a lot of jumping around. Which strand is going to get done first? On my paper, let's just go with left and right. Which strand is going to get done being made first?
The right one.
The right one. So in a race, isn't it in the lead? So they call the one that is continuously made the leading strand. Okay, they call it the leading strand. Hear those terms again. Okay. All right, so the leading strand. The one that is made afterwards is the lagging strand. Isn't it lagging behind? Okay. And on this lagging strand, don't we have a bunch of pieces that aren't really joined together yet? Those pieces are called the Okazaki fragments. They're called, it's named after a guy, Okazaki fragments. Okay. So this one was continuously made. This one we really don't have to do anything with. It's kind of done. Here we've got some work to do. So now what has to happen is this. We've got to get rid of those primers that Megan's been wanting to get rid of for a while now, right? There is an enzyme called DNA polymerase 1. So now we're looking at DNA polymerase 1. DNA polymerase 1 is going to come in. It's going to remove the primers. So it'll go and it'll move this primer, and it's going to add DNA to it. So we got rid of that primer, so now can't we put a G there? Okay. Let's get rid of this primer. DNA polymerase 1 comes, and it gets rid of this primer, and we'll put a DNA base in there. It gets rid of this primer down here, and now it can add the DNA nucleotides there. Now we've got all DNA nucleotides, correct? But don't we still have a bunch of pieces that aren't joined together? Like this fragment was never joined to this fragment because there was a primer in the way. And now we put DNA, but you still have to join this to this, and this to this, and this to this, and continue on. We have to join all this together so it's continuous. What type of bond needs to be formed to make this strand continuous? What type of bond needs to form between this guanine and cytosine nucleotides? A strand. Phosphodiester. There is an enzyme called ligase. Think like ligatures, right? See the LIG, like ligatures join things together, okay? Ligase will join. It's gonna come and form a phosphodiester bond here, okay? So phosphodiester bond by ligase. It'll join here and make a phosphodiester bond, a phosphodiester here, a phosphodiester here, a phosphodiester here. So it'll fill those gaps in. Now, don't you have a continuous strand? Right? Okay. So now what you would basically see is this. You've got 2 DNA molecules. The black strands are the original strands we started with. Didn't we start back in that original picture with, 2 strands of DNA? And we kept separating the strands out, right? The 2 strands, black strands, were your original template strands. Didn't we make the new strands on the inside? So here's a DNA molecule, here's a DNA molecule. How do they compare to each other? Look at that. Aren't they identical? Didn't you just make a copy? Okay. This type of replication where you end up with a DNA molecule that has like an old strand, I'll just say old or template, and then it's got the new strand. Here's the old, here's the new, right? Doesn't each DNA molecule have an old and a new? That is called semi-conservative replication. Okay, it's called semi-conservative replication. It's not all new, some of it's old, so it's semi, okay? And once the DNA strands are made, this just winds back up as a double helix. Now you've got 2 DNA molecules. If you get rid of color, well then here we go. We've got 2 DNA molecules in the end. 3', 5', 3', 5', right? And that's what we were going towards.
Can you repeat semi-conservative?
Semi-conservative replication is where when you're making a DNA, new DNA molecule, you are using an old strand, a template strand to make a new strand, a template strand to make a new strand. So that in the end, your DNA molecules have an old strand and a new strand. Old strand or original or template strand and the new strand.
Thank you.
And then what just happens is basically now you continue on. This DNA becomes the template and you make 2 new DNA molecules. This DNA becomes a template and you make 2 new DNA molecules, which is how the DNA in your body is all exactly the same. You're just making a bunch of copies, okay? Don't forget that you've got the, sugar and the phosphate here and the sugar and the phosphate here. And you do have a phosphodiester bond that forms between them, right? Okay, so you do have a like 2 strands, double helix. Now we can go through this process, and can you imagine 6 feet of DNA and 3 billion base pairs and making a copy of that and taking 6 hours? Nothing's perfect. So what happens when this doesn't go perfectly? Like, for example, adenine is a purine. What other base is a purine?
Guanine?
Guanine. Purines have a double ring in their structure. What if by accident a guanine was put here? That's not correct, but it fits. They have a similar structure. It was a mistake. You can't explain a mistake. It just happened. So what has to happen in the end is an enzyme called nuclease. Nuclease is a proofreader. Kind of comes along and like, oh, good, good, good. And they're like, oh, crap, there's a mistake. So nuclease is going to cut out the mistake. It's going to get rid of that guanine. It's going to cut it out. Okay? What will add the correct base in there? What will put the adenine in there? What enzyme have you seen that adds DNA nucleotides? All it does is add DNA nucleotides. It makes
Polymerase 3?
DNA polymerase, and it could be 1 or 3, really, okay? But we'll just say DNA polymerase. DNA polymerase will come and add the new DNA molecule. Nuclease finds the mistake, cuts it out, DNA polymerase corrects it, okay? So there is that proofreading in the end as well. Remember we went through and we talked about, do you remember talking about checkpoints in the cell cycle? And there's checkpoints to make sure spindles attach properly. There was a checkpoint to make sure the DNA replicated properly, okay? So there's checking. Now, mistakes can go unnoticed. And if that happens, what if nuclease missed a mistake? What would that mean?
Mutation?
A mutation, okay? And we'll be talking about mutations in second semester because that also leads into evolution and everything, so, okay? All right. How many of you, by just, let me like see you all first. Okay, so turn your cameras back to this. Okay, how many of you feel like you at least were able to follow along? Just kind of raise your hand so I get a quick glimpse. Quick glimpse. Rocco's been like talking and texting all period. I don't even know what he's doing. Okay, so I'm gonna put this recording. Oh, you can put your hands down. Okay. I'm going to put this recording online. If you were able to at least like sort of follow along and you're like, okay, I was able to follow, I just need to sit with it now and just make sure, then you're in a good place. Then that's good. Okay. So I'll put the recording on. And also, what time is this class over?
05, I think.
Okay. So I'm just going to show you, screen share to show you this video. I'm fast forwarding through the video so you can see it, but let me screen share. Okay. So what you're going, I put this link online as well. Okay. So take a look at this link. And like I said, I'm just pushing through it, but here's your DNA molecule labeled 3', 5' anti-parallel strands, right? There is an enzyme, this green enzyme. What is that enzyme? Look what it's doing already. What's this enzyme?
Helicase?
Helicase, isn't it separating the 2 strands? So helicase is pushing through and separating the 2 strands. Small segments at a time though, okay? And as it does that, you'll have DNA polymerase, this would be DNA polymerase 3, making a new strand 5 to 3. The one that is continuously made, what is that strand called?
Leading strand.
Leading strand. So look at that strand, it's just continuous. On the other side, though, there has to be an RNA primer that goes in place so that DNA polymerase 3 knows where to start. And so in actuality, I showed you in steps, but in reality, ligase, DNA polymerase 3, DNA polymerase 1, they're all working all at the same time. It's not like, okay, you finish it all and then I'll go do this. It's like, if you're already like 2 feet ahead, why don't I work from where you just left off, 2 feet behind you, and start there? Why wait, right? And so again, remember, this is 6 feet. This is not a sheet of paper like I showed you, 6 feet. So once DNA polymerase 3 has done its job and moved forward, we can start filling in the gaps. So you'll start to see the lagging strand being made and RNA primers put into place. So RNA primerase comes in and puts a primer. The pink is a primer. And so that tells DNA polymerase 3 exactly where to go. So it goes and makes a short segment, right? 5 to 3. And again, this is all happening at the same time. So once that part is done, DNA polymerase 1 can come in, get rid of the primer, okay? And you'll just see that continuously being made. And then ligase can fill the gap, make the phosphodiester bond. And so you can go through this slowly, but RNA primase puts a primer in pink. DNA polymerase 3 adds a strand, short segment. And so you'll see this process just taking place. And look at the leading strand is already almost done. And so they label the things for you as well. This link is already on Schoology. You can just Google also, okay? But, and find other stuff to help you, but I did put this one, okay? So that's where we're ending as far as semester material goes. You should be able to do the optional homework for Monday and the FRQ as of now, if you don't want to wait and you got a lot of stuff. So I wanted to try to get that done so you have the weekend to be able to do work. Do you have any questions before you go? Because it's pretty much time for you to go.
No.
Okay. Reach out to me if you do, okay, because I don't see you tomorrow. People that have been emailing me about your grades and stuff, I've been updating as I go, okay? So as soon as I change your grade in the computer or delete something that you didn't want in there, I usually will respond back to you and just say done, okay? So double check that stuff. Those that haven't done 4 FRQs, you've got 1 more left, okay? So please check on that. And with that, I'll let you get to your class. Get ready for your next class, and I will see you on Monday then, okay? Again, you have everything you need for your test, right? All right, and I'll put
Thank you.
Thank you.
Thank you.
Bye, guys.
Thank you.